I'm Bob Waterston, the chair of Genome Sciences and Gates Professor, and I'd like to, first of all, welcome you here on behalf of the Department of Genome Sciences. I think we have a wonderful evening in store for you. I'm going to try to get out of the way pretty quickly, but we're going to be discussing, as you know, the personal genome, consequences for society. I think we have just an outstanding panel tonight. And we have a topic that's obviously of wide interest. So, as many of you know, genetics is in the midst of a revolution. Over the past 15 years, we've moved from being able to study just one gene at a time to be able to study all the genes of an organism. That is the genome of an organism. This revolution, I think, was initially driven by the Human Genome Project, which was a project to get the sequence of the human genome, that is, all the genes of humans. But more recently, geneticists and genomicists, as we're now calling ourselves, we've been using that sequence to decipher genetic contribution to human traits and diseases such as diabetes, cancer, and heart disease. And as you'll hear tonight, we're on the verge of new technology that's on the horizon, I would say, that will let each of us know our own genome sequence and the variations that distinguish us. This is very powerful information, and like any powerful information, it can be subject to misuse. Societies have not always done well in harnessing such powerful information for the public good. And in genetics in particular, the eugenics movement of the early 20th century, a combination of prejudice and bad science, provides a telling example of how things can go wrong. So one way to help ensure a good outcome with this information is through openness and the involvement of the public like you. We need an active, informed public. The Department of Genome Sciences is trying to do its own small part in this. We've been strong advocates of free, open access to the data, basically public ownership of the data. We're going to begin with a short background talk by Eric Lander to give context for the discussion that will follow. Each of the other panelists will then say a few words highlighting a topic or concern that's of special interest to them. But the bulk of the time will be devoted to the panelists discussing questions that you submit. Provost Phyllis Wise, on behalf of the university, would like to say a few words of welcome. Phyllis.
Well, I too will get out of your way really quickly so that we can get on with the evening, but on behalf of the university, I really want to welcome you here this evening. This is sponsored by the School of Medicine and Genome Sciences, and this is certainly one of the departments that we are most proud of. It was founded in 2001 through the combination of the Department of Genetics and the Department of Molecular Biotechnology, and has some of our most preeminent faculty, members of the National Academy, Nobel Prize winners, MacArthur awardees. It is truly the kind of department that we feel allows the University of Washington to do the kind of revolutionary science that it does. And as a biologist, I have to say that I've never been in a room with 4 people who have so changed the way we think about life, so changed the way we practice medicine. I think we are incredibly honored to have the 4 of you here. And so I want to welcome you and look forward to the evening along with everyone else. Thank you.
Thank you very much, Phyllis, for the kind words. We're trying in genome sciences. So now it's time for our briefing from Eric Lander. Eric is professor of biology at MIT and is the founding director of the Broad Institute in Boston. Eric received his BA from Princeton and his PhD from Oxford, both in mathematics. He then joined the Harvard Business School in 1981. But in the mid-'80s was lured into the Human Genome Project. At that point, it was not really a reality, but he got interested in it. By 1990, he moved to MIT Biology. Eric has been a leading figure in genomics ever since. His Genome Center at the MIT Whitehead and then at the Broad has been a major contributor first to the sequencing of the human genome, then the mouse and many other genomes. And in more recent years, he's focused his attention increasingly on human sequence variation and its relationship to human disease. Eric serves on various advisory and governing boards of several institutions. I'm not going to list all these, and I'm not going to list all the honors and awards that he's won. But I'll mention two. In 1987, he was named a MacArthur Fellow. And in 2002, he shared the Gairdner Award of Canada. He's also a terrific teacher and lecturer, as you're about to find out. So without further ado, Eric, pleasure to have you.
Thank you, Bob. So it's a real pleasure to be here and just try to give a little bit of a backgrounder on what our discussion will be about today. At MIT, I teach freshman biology, and I'm going to attempt to compress freshman biology into about 15 minutes, at least the part of it relevant to the personal genome that we're going to talk about. And so let's dive right in. There will be a test at the end. I'm going to try to cover the period from about 1980 to roughly 4 o'clock this afternoon, if I can, and give you a sense of how things have changed. So look, before 1980, the way people approached the study of disease is you needed some inspiration. You needed some particular guess as to what to look for. What really began to change, and what excited human geneticists a great deal, was around 1980, the suggestion that you could start being systematic, that you could approach diseases in a systematic way regardless of what kind of disease they were. The idea was this. It was that if a simple Mendelian, that means one gene disease, was being passed down in a family, as it is in this picture here from Dad, who's a colored square there, to 4 of his children, then if you wanted to find out what that disease gene was, you should instead ask where it is. And the way to ask where it is try random DNA spelling differences up and down the genome until you find one, the one that's indicated there, where Dad got an A from his own dad and a C from his mom, and he passed on the C to all the kids who got the disease. There's a correlation there between the inheritance pattern of the disease and the inheritance pattern of this spelling difference. And if you saw that not just in one family, but in enough families, you'd be able to conclude that the disease gene, whatever it was, must live nearby on the chromosome to that spelling difference. Now notice I didn't tell you if it was a brain degeneration or a bowel disorder or a heart disease, because it doesn't matter. It's the same basic idea. You can find out approximately where something is. Once you know approximately where something is, you could march along the chromosome to actually get to the disease gene. I remember in 1985, the day I heard that cystic fibrosis was linked to a particular genetic marker on chromosome 7, and it was 99% correlated. Of course, 99% correlated meant 1% uncorrelated, which meant 1 million letters of DNA away. And a tedious process over the course of 5 years involving tens of millions of dollars and more than 100 people led eventually to the cloning of this one important disease gene. And its sequence was read out, and there it is. It's a boring string of A's, T's, C's, and G's. But I call your attention to the little red box, because those 3 letters in the little red box are deleted in the vast majority of individuals who have cystic fibrosis, and about 1 in 20 of you are carriers for that mutation there. Now that's important. You could do genetic diagnostics. More than that, you could take the sequence of that gene, toss it into the computer, ask the computer to compare it to every other gene that's ever been found, and say, does it look similar? And the computer immediately comes back and tells you, ooh, the gene you just found looks a lot like transport proteins. It's probably a transporter. And so you get handed a good biological hypothesis. So this is pretty exciting. You didn't have to know what the disease was about in the first place. You map where it is by randomly looking for correlated markers. You sequence it. You can do diagnostics. You can look up the function. What more could you want? Well, what more you could want is that, of course, this one disease gene took 5 years, tens of millions of dollars, 100 people working on it. What young student would want to sign up to be an ant in that army? And we know that the unit of progress in science is the smart young student. The goal was to make this tractable for a single student to be able to do this by him or herself, and that was the purpose of the Human Genome Project. It was biology's first great infrastructure building project, to collect that information, to collect genetic maps of those spelling differences up and down the chromosomes, the physical maps of those overlapping pieces of DNA, the sequence map of the underlying letters, the gene lists in that sequence map, and very importantly, and Bob played an extraordinary leadership role in this, ensuring that all of that information was freely and immediately available around the world to anybody, in industry or academia, in developed countries or developing countries. It required changing the way we do biology in all sorts of ways, industrializing aspects of biology so that the work didn't have to be done by legions of graduate students chained to the bench pipetting. Robotics were developed both in academia and in industry, better sequencing machines, so that one could generate millions and millions of bases to eventually be able to get a 3 billion letter genome covered enough times that you could assemble its sequence. It involved international collaboration of a sort that hadn't happened before in biology. 20 different groups working in 6 different countries, the United States, the United Kingdom, France, Germany, Japan, and China, labs of very different scales, taking different approaches, but all committed to this common goal. It led, in February of 2001, to the publication of a rough draft sequence of the human genome, still imperfect. Only about 90% of the sequence there, lots of gaps, lots of mistakes here and there. But this international consortium continued on, and by April of 2003, produced a finished sequence of the human genome. I always, whenever I say that, have to put in the footnote, there is still some bits of the human genome we can't quite sequence, centromeres, but for our purposes, we'll ignore that right now. We have a finished sequence, but finished is a technical term that we work very hard to define. So, it means everything we can do with existing technology. It's a beautiful genome. Your genome is laid out there, and you can see regions that are gene dense and gene poor, and you can see regions that have lots of G's and C's and less G's and C's, and how they all, you can see it's like flying over a landscape of the United States and seeing the Grand Canyon and the Rocky Mountains. All these features that you can see only by pulling back from a distance become apparent. But the question was, did it make it possible to find those simple Mendelian disease genes, those single Mendelian disease genes that were so hard to find in the late 1980s? And the answer is yes. Before the Human Genome Project, only about 70 of them had been found. With the Human Genome Project, with the advent of that and the information and technologies, that number has grown to 2,600. And if one graduate student at UW wanted to find the cystic fibrosis gene today, imagining it hadn't already been found, it would be a perfectly good rotation project for someone a few months in a lab, which is just fantastic. But it's not enough. Because those are the simple Mendelian diseases, mostly things you haven't heard about. Diastrophic dysplasia and other, complicated names and things. What about diabetes? What about asthma? What about heart disease? What about manic depression, schizophrenia? Well, we know that those are not caused by simple single genes, by simple Mendelian transmission in families, and this method didn't work very well for them. So we had to move onward to the question of how were we going to find the genes underlying common multigenic diseases? And for that purpose, it was important to step back and understand the genetic variation in the human population. I'll just briefly describe the genetic variation in the human population. If I take anybody out of this audience and sequence a stretch of your DNA from your mom and your dad, or if I take two genomes from any 2 people in this audience, you will differ by only about 1 letter in 1,000, actually a little less than that, 1 in 1,300. That's the extent of variation in our population, only about 1 in 1,000. And if I sequence you and I get a site of variation, a site of heterozygosity, there's a 95% chance that that site of variation is shared with other people in this room. The vast majority of the variation that you've got is worldwide common shared genetic variation. And so, even though you have 3 million sites of variation in you, when we start adding everybody up, the number of sites of common genetic variation in this whole audience, in this whole world, is only about 12 million. Now, skip over that. Some of those sites of genetic variation matter. Here are 2 letters that are variable in chromosome number 19. It could be either a T or a C at the first position, and a T or a C at the second position. If you happen to have gotten the CC spelling from mom and the CC spelling from dad, you have about a 60 to 70% lifetime risk of Alzheimer's disease. And about 3% of this audience does have that. And if they spit in a tube on the way out the door, Bob could take it up to a lab and do an amplification and send you an email tonight about whether you have that. Now, technically, it would be trivial. Ethically, it would be complicated. He couldn't actually do that because the counseling involved, et cetera, these are serious issues. But technically, it costs $1 to do something like that. It's not a very big deal. And we'll come to questions like this. But the truth is, we only knew a couple dozen examples of these common genetic variations in a population relating to common diseases in the population. Not good enough. So around the mid-1990s, this idea began circulating amongst human geneticists. What say we just collect all 12 million genetic variants, write them down along the top of one very large Excel spreadsheet, and then write all the diseases down along the rows of the spreadsheet, and figure out which variants are enriched in which diseases. People who have one variant, people who have this variant are more likely to have diabetes. People with arthritis are more likely to have this variant. And that big spreadsheet, 12 million genetic variants by all the diseases, we could call human genetics. That would be a description of common disease, perhaps, if this idea would work. Now, the idea was certifiably insane in the mid-1990s because, of course, at the time, the number of genetic variants we had numbered in the couple of thousand. The thought that we'd ever get to 12 million was crazy. And the thought that we'd ever be able to, even if we got to 12 million, genotype them in each person, that would be, if we wanted to do a study for diabetes, it'd be thousands of people, millions of genetic variants. How are we going to do that? It would be billions and billions of genotypes. But science works by putting one foot in front of the other. And like with the Human Genome Project, that in its own day was considered pretty outrageous, well, you start going. And, whoops, wrong way. By 1998, methods using DNA chips were developed to begin to collect more genetic variants. And then other methods were developed that led to the creation of the idea of a consortium, an international consortium, to be able to find more and more of these genetic variants. The goal, which sounded pretty stretch at the time, was 300,000 of them. In fact, it proved to be underambitious because within a year, the goal was blown away and 1.4 million of the genetic variants were identified. And Bob's center and my own center were part of this effort, and it was very exciting. It grew to 2 million, and 4 million, and 8 million, and 10 million. And today, if I go to your genome and I get a genetic variant out of your genome, there's an 85% chance it's already in the public databases. That's the extent of coverage of our knowledge of human genetic variation today. Now still, the prospect of testing those millions of genetic variants is not very attractive. I mean, especially because graduate students used to do it one at a time, so this is really very unattractive. So maybe we don't have to test them all. Maybe the ones that are nearby are so well correlated that we don't have to worry about that. And so ideas grew up to look at the correlation patterns of genetic variants in the genome. And around 2001, some interesting observations were made about the nature of correlations of nearby genetic variants that suggested that maybe if we knew that whole pattern, we could get away with only using a few 100,000 genetic variants. So again, a crazy idea was proposed. Let's make an international project to find out all the correlations of genetic variants in the genome. Folks argued about that for a year, agreed to launch the project in 2002, completed the project by 2006, and we now know the correlation structure of genetic variants across the genomes, which ones are well correlated with each other's, where the correlations break down. Now, you only have to do 300,000 genetic variants. But that still sounds like a lot of work. But technology coming to the rescue. The idea of doing genotyping one at a time yielded to technologies to multiplex them and do 10 at a time. And then these DNA chips to do them 1,000 at a time, and then 50,000 at a time, and then in 2006, 500,000 at a time, and by last year, 1 million simultaneously. So suddenly, the crazy idea from 1996 was actually quite doable. You could take 1,000 people with diabetes, 1,000 people without diabetes, look at 1 million genetic markers in both, and make the spreadsheet. And so 2007 was a period of great anxiety for those of us in human genetics, because either this was going to work or it was going to fall on its face. And I'm happy to report that 2007 was an annus mirabilis, one of these fantastic years in science when things just break. Here, I'll give you an illustration. This is going to show the genetic variants associated with common diseases that were discovered In 2000, 1 for diabetes. In 2001, 2 for Crohn's disease. Nothing in 2002, but in 2003, 1 more. It's poking along at about 1 a year. 2005, 1 more for macular degeneration. 2006, those tools are just beginning to come online, but not really available yet. 5 were found. 2007, through the month of April, through the month of August, through the month of September, and I don't have the rest of it, but it's another 2.5 columns that run off the page there. There are about 160 associations between common genetic variants and common disease that have been found just in the past 18 months. So this is very good timing for this symposium. What are they telling us? Well, for people who study any particular disease, it's handing us all sorts of clues as to what's going on in that disease. But what are the take-home messages for you in this discussion? Key lessons. Number 1, the stuff works, but to make it work, it's taken large samples, thousands of people, and stringent statistical thresholds. 10 to the minus 8th significance, if you know what that means. If you don't impose stringent thresholds, you get all sorts of blips and could get excited about them. But this stuff holds up now to real scrutiny, and it's reproducible when you look at large samples. We have only scratched the surface. As excited as I am, what's very clear about this is we know only a tiny fraction of what matters so far. We've looked at only a small number of diseases, and even for those diseases, we've only looked at certain types of genetic variants. So the story is just at its beginning stage. These variants that I'm telling you about, these 31 variants for Crohn's disease, their effects are pretty small. They might increase your risk by 10% or 20%. But to a biologist wanting to understand the pathways involved in Crohn's disease, couldn't care less how big the risk is. That gene plays a role, it'll point to a pathway. But to a patient wanting to understand, does the fact that my risk of diabetes is now 10% higher because of that gene mean anything? Well, your background risk of diabetes was 8%. If you have a variant that increases it by 10%, it's now 9%. I don't know that you're going to do anything very different because of that. Although you might start suddenly saying, oh, I'm diabetes prone. Although it's not clear that there is any meaningful notion to that. So we've got these two things in our head. Biologically, this is incredibly important stuff. It's going to remake our understanding of the disease. From the point of view of a patient, me as a patient, I'm not sure any of this is anything I'd act on. The effects are still small, and many of these things we don't really have an action for anyway. Beware the flood. There is an increasing flood coming behind these really great studies here with p-values of 10 to the minus 10th that are reporting much more dubious sorts of things. Now, the one other aspect of our genome that I must mention before stopping is the cancer genome. We've talked up to now about inherited genetic variation, what you get from your parents. But there's another kind of genetic variation, which is the mutations that happen in your own body and give rise to cancers. You could ask, why don't we start knowing them too? Well, similar work has shown us over the course of decades that cancer is a disease of genetic mutations as well. It is driven by mutations, and studies in the 1990s and early 2000s have identified by individual sequencing of genes or sets of a few dozen genes, particular genes that are mutated in lung cancer or in breast cancer. But the real question is, why not have the whole menu laid out? Why not do the same thing for cancer? Lay out all the mutations that could occur in cancers so you have a comprehensive menu. When someone goes into the hospital, they can have their tumor sequenced and understand what the mutations are and correlate it with how they may respond to different therapies. So in 2005, Lee Hartwell here in Seattle and I co-chaired a committee that suggested a Human Cancer Genome Project. And the National Cancer Institute and the National Human Genome Research Institute have actually just launched such a project last year. And the first results of it are just coming in these past couple of months, and it's very exciting. We're already seeing that huge curve ticking up. Lung cancer, we thought we knew the genes. Well, there are 4 or 5 commonly mutated. That list will triple before the end of the year. Glioblastoma, that list will double by the summer, etc. Now, the last point, the road ahead, for one of my panelists here. Everything I've talked about is with existing technologies. But if you talk to genomicists around this building, this Department of Genome Sciences, what they're really looking forward to is the new technologies that are coming on. Powerful new sequencing technologies that make everything we've done before look pretty puny. The Human Genome Project was done with machines somewhat like the one on the left. Not quite, actually, an earlier generation of the machine on the left. And on a great day, you know, on a great run from that machine, you might get 70,000 letters of DNA. But in the past couple of years, machines have come along that can produce 100 million letters of DNA. A new one, 2 billion letters of DNA in a run. A new one, 10 billion letters of DNA in a run. This is not totally a kosher comparison because some of the runs are longer than others, but anyway, you get the point. It's extraordinary how this is exploding. And the cost of doing a human genome, well, the first human genome, this Human Genome Project, a 15-year program to develop the technologies and apply it, was $3 billion. It fell to 5 or 6 years ago to about $30 million to do it again. To today, probably about $300,000 to do it. And the gleam in everyone's eye is the famous $1,000 genome, which can't be done. We can't come close to it today. But it's not crazy to imagine. And what this is promoting now is a world of enormous scientific possibility, but also reaching out into the general public. And this is why the university here has brought together the public in these great public education and public discussion fora, because this stuff is coming direct to you already on the web. There are numerous companies that have been put together. I've put up the names of some of them, 23andMe and Navigenics and Decode and Know.me and DNA Direct, where you can spit in a tube, and they'll do those million markers on that chip that I told you about. And they describe what they're doing in very different ways. One of them says on its website, your genes offer a roadmap to optimal health. Maybe, or maybe your problems and your solutions aren't found in your genes. Who knows? That's your backgrounder. On to discussion. Thanks very much.
Thank you. Thank you very much, Eric, for a wonderful talk. I knew you'd do it. And so now on to the heart of the program, the panel discussion. And joining us to moderate the panel is our own Maynard Olson. Maynard is professor of medicine and genome sciences. He was doing genomics before there was such a thing. He was a key witness and then a committee member of the National Academy Committee that produced the report that provided the framework for the Human Genome Project. He too was a major contributor to the sequencing of the human genome and other genomes, and he too has pursued human variation more recently. Among his awards and honors, he shared in the Gairdner Award of Canada in 2002, and this past year received the Gruber Prize in Genetics. His sage advice is keenly sought by me all the time, by scientists and institutions and governments across the world. I could go on, but I know people are anxious to get going. So Maynard, the floor is yours.
Well, thank you, Bob. It's really exciting to have this panel here and such a great audience. And we couldn't have a better group here. So you've already met Eric. Next to him is Leona Peltonen. She's the director of Human Genetics at the Wellcome Sanger Institute in Hingston, the UK. She is a true international person, Finnish origins, has worked extensively in Finland, which has a really fascinating sort of human genetic heritage, has worked extensively in the United States, and is now really one of the foremost institutes doing the type of work that Eric was describing and heading their Human Genetics program. So she can sort of tell us where is the state of the art today and how are these more futuristic elements going to play in. I think Bill Gates needs no introduction. There are numerous ways in which his unique background is relevant to tonight's discussion. Here's someone who understands technology and its transformative effects on society and knows how to take seriously rapid technological change that is sort of pushing applications unimagined 1 year out into common use in a few years. And of course, another side of Bill's activities, which are well known to you through the Bill and Melinda Gates Foundation, he has a really fundamental interest in how high technology, the best science, and anything else that we can figure out how to do can be brought to the benefit of the whole world, and particularly the world's poor. I just would say one more thing about Bill is that we are in great debt to him at the university for having this extraordinary program in genomics, and I personally benefited enormously from his visionary gift in 1992 to form the molecular biotechnology program. It's what led to my recruitment to the University of Washington, and was sort of the foundation for a really fabulous 16 years in a premier environment. And then at the end of the line is George Church, who's been one of the primary, I would say the primary, sort of intellectual thinking about personal genomics from both a technical and a social point of view. I first met George when he was still a graduate student, I think about 1984, and already this sort of tall, distracted-looking graduate student had a passionate idea about sequencing genomes right there on the lab bench that was in front of you. And this passion has actually not changed. George was really not much to be seen during the industrial phase of genomics. Industrial activity is not his thing. He wanted to be able to sequence genomes today on a lab bench cheaply, and to push this technology out into the real world and where we can actually deal with the complexities in front of us. So since that time, he has pursued his career at Harvard Medical School, where he's now a professor of genetics and a leading spokesman for the issues surrounding personal genomics. We will now move ahead and start with a question for all of the panelists. Should people be told about their risks for genetic diseases that have no preventative measures and no treatments? Eric?
Should they be told whether they want to know or not? Is the which one?
Both.
Should they be told against their will? Absolutely not. Should they have the right to find out? Absolutely. How are they going to get the education to be able to make an informed choice? That's really a hard question.
Lena, you want to try this?
Oh, of course, what else can you do except agree? But I think it's a huge challenge that how do we translate this massive amount of information like Eric elegantly described we are collecting. So how do we translate that new information to knowledge or understanding, and even more to some level to medical practice, or to practice to provide information to people that they know what it really means. And it's a huge challenge, and perhaps this is one of the events we should discuss those issues. How do we handle that?
I mean, I think it's clear that that no one's advocating telling people against their will, but the core issue is what the standard of care should be. Mm-hmm. You know, once, say, cholesterol testing above a certain age became the standard of care, then that information was very widely available in the primary care environment, and most people will then be exposed to it. There may be occasional ones who don't want to hear it. But George, did you have?
A small point, which is if the key thing is actionability. If it's not actionable, there is something that you can do, which is you can become an activist. That is to say, you can go out, get yourself educated, educate your community, raise money for it. If there are some end-of-life decisions to be made about exactly how you're going to have to live and where you're going to have to live, that might be good to know that earlier rather than later. So everything is actionable in a certain sense.
Good. Let's take another. People had the option of identifying themselves. This one came in over the internet from Sally James. Thank you, Sally. Given all the influence of our environment on our health, doesn't the public overestimate the power of genes? I see Lena nodding.
Yeah, definitely. But we should also remember that this information is, not yet there. I think like Eric described, so all these tools were actually developed to produce more understanding and more scientific hypotheses to be tested. They were not created to serve as a diagnostic setting. So I think it's a huge difference how we tackle this information and how population-specific or family-specific. Marie-Claire King has beautifully shown how valuable for some families or some population is the testing of very rare variants predisposing to breast cancer. And still the common breast cancer variants are still pretty much in the dark. And the value of the information you obtained in specific, if you are part of a specific family or you are part of a specific population, the value of this information is completely different. And what slightly worries me actually is that there are currently about 500,000 people on the globe whose genome is pretty thoroughly scanned with these chip technologies. Less than 20,000 of those are something else than Northern European origin. So our understanding of the landscape, which Eric elegantly described, like we look from the plane, where are the canyons and where are the rivers, that landscape is different for different populations. And we know next to nothing of that landscape for Africans, for Asians, for Southern Americans, and that's an issue we need to tackle as a scientific community before we start selling these Decode Me tests.
If I can jump in, and I agree with what Lena said, I really want to pick up on the notion of the power of genetic information over other kinds of information. Mm-hmm. I think that the public, all of us, probably me too as a patient, overvalues genetic information. I think we do treat DNA evidence as definitive. And of course, it's incredibly important. I hope I convinced you of that. It's also no more definitive in many cases than many of the other influences. And so getting an understanding of what DNA is telling us, a proper understanding that values its real biological information, but also puts it in its place, is going to be a huge job. And I don't think we currently are close to that.
So here's one from Jenny Hannibal, which maybe we'll start with Bill Gates. It's an interest of his, as of all of ours. The personal genome is likely going to benefit only those in developed countries. What do you see as the options for this to benefit third world populations, and what can we do now to ensure that it happens?
Well, if we define personal genome fairly narrowly as rich people in the next decade getting their genes sequenced, that won't have too much benefit. Anytime you divert, say, $1,000 away from spending on health for the poor, you basically lose a life. That is, we can save lives for about $1,000 by things such as improving the vaccination systems in developing countries. But if we define personal genome differently, if we define it as the making genetic information almost free for scientists to gather, then the benefit of this revolution for health conditions in the poor countries will be very, very dramatic. So we need this information for diagnostics, and I'd say by far the biggest beneficiary of this will be the greatest unsolved challenge in global health, which is the deep inequity that young people still primarily die of infectious diseases, and it's not really because of genetic variation, but genetic understanding will let us create cures that we should be able to get rid of those 20 infectious diseases, the top 20 infectious diseases.
I'd like to just combine this with the previous question about genes versus environment. And the point of this technology, which is exponential, maybe even faster than Moore's law that we see in the electronics and computing industry, this is a very fast changing technology, is going to impact our ability to sense our environment. Our environment consists of microorganisms, viruses, allergens, and so forth, and there's no reason why this technology can't get down to the point where for less than $1 in developing countries, and probably quite a bit more in developed countries, you can do simple types, multiple assays in one go. And I think that's where we should be thinking about this going.
So here's one topic that I know George has written extensively about, is how will personal genomics affect the privacy both of initial research subjects and of everyday consumers, especially if they're revealed to have an increased risk of disease?
There may be people who voluntarily decide to give up some of their privacy, and that shouldn't necessarily set an example for other people. It should be an individual decision. There needs to be a great deal of education. Has to be very strict testing to make sure people know what they're getting into in every sense, and we need to monitor that with time. Research subjects are beginning to ask for their data back. About 90% of them want to know about themselves. They consider it immoral not to do that. That's the path to loss of privacy in a certain sense. So I think we need to go forward very carefully and not necessarily consider that people that are doing research are the same as people that are going to these companies that Eric showed. That's a different thing. You should keep that private until you know what the consequences are.
So just a follow-up question would be that if one sets the informed consent, especially the informed part of the informed consent bar, too high, which some would argue it presently is, then you end up sequencing the genomes of prominent geneticists and other highly atypical representatives of humanity.
Useless.
Is there a solution to that problem?
Well, first of all, I'd like to point out that there have been 2 peer-reviewed genomes, but there have been announced 2 African genome sequenced and 1 Chinese. So it's not quite as white bread as it sounds.
But I don't believe those people are identified.
That's true, they're not identified. But I
The
don't
2
think,
geneticists were.
yes.
Yes.
I think it's an open question as to who's. So far in the Personal Genome Project, we're seeing people from a whole variety of ethnic groups and socioeconomic status, but I think this is an open question as to whether this scales up into the hundreds of thousands that we need in order to get good statistics.
When you do consent to reveal your genome, do you get all your relatives to also sign the same document?
That's an excellent idea. Modulo.
But it don't happen, no.
You're revealing their information.
Of course.
I mean, that's not
Your
fair.
kids especially.
Your future kids particularly.
Right.
We do, in the Personal Genome Project, we have eliminated some people who had relatives that were not okay with it. So there is a component of that. The IRB did not require it, but we think it's a good idea going forward. It's all a matter of balance, as Maynard is saying, as to whether it will exclude too many people.
Here's one from the live audience. Does knowing about all of these DNA variants not risk a world of designer babies?
I don't think anybody thinks so. At least if you are a geneticist, you don't think so. Let's think, some of these common variants which have been associated with type 2 diabetes or obesity or high lipid values, which are considered risk, like bad for you. And in the tailor-made babies, you perhaps would like to eliminate those. So from genetic point of view, it doesn't make any sense because they are so prevalent in our genome, as a community, because they have served like wonderful benefits for our ancestors. Human species have survived because they were able to efficiently digest lipids. So it would be a very short-sighted policy from genetics point of view to eliminate certain variants of human genome. Mutations are good. Mutations are fuel for evolution. So from that point of view, it doesn't make sense. And also, let's put it aside, it's impossible to modify genome at the level of accuracy you would like to perhaps see to happen, or modify ApoE4. But currently, I would be strongly opposing that, because we don't know the consequences. Let's assume 1,000 years from now, it would be highly beneficial to have ApoE4 for Alzheimer's for an unknown reason. Our environment changes or whatever. We can't judge that.
Are there other comments on this?
I'll take a slightly different approach I guess the question whether we should do it or whether it will happen. Because if you flip it around to whether it will happen, even if we shouldn't do it, you see a lot of parents that are doing everything they can to coach, purchase coaches for their kids. And if we're talking here not about the whole world, but the elite, there will be some tendency to do that. And also it depends on what you mean by designer babies. Do you mean that you're actually changing the germline, or that you're actually fixing them as they grow up? Because in the second category, we already do that.
Oh, that's a different thing. I meant germline.
But I'm not sure what the question is, because there are a lot of kids that we're fixing that aren't necessarily broken. Kids with mild ADD, or just taking their friend's Adderall just for fun, because they think it'll get them higher grades, okay? This is designer babies, and we're doing it.
Or look at the orthodontic industry as flourishing.
There you go.
Yeah, if I could just ask a follow-up question on that. The question, of course, asks about, is there a risk? And I suppose that does mean, do you think this is going to happen, and will it be bad? Lena's expressed her grave reservations about the badness part of it. George has indicated that in a world where people make up their own minds, certainly in our society, we just tend not to tell people what to do. They certainly will do it. But let's make the specific context of, to be slightly technical, pre-implantation diagnostics in conjunction with in vitro fertilization. So just very briefly, this would be the most high-tech, I would say, sort of eugenic procedure that we now know how to do. So an embryo is created in vitro by in vitro fertilization. After a couple of cell divisions, it's been shown that you can remove one cell without damaging, at least supposedly. We'll find out what happens when these kids grow up. This has only been done for a few years, but it appears not to damage. Subsequent development, genetic tests can be done on the genome in that one cell, and you do this on a whole series of embryos, and you pick the ones that are either free of a defect, or I think what is really behind this question is that we will learn about genetic variants that predispose to traits that are highly admired, say physical attractiveness. I think it's not safe to assume that the genetics are going to be so complicated that we can't untangle such issues, and people will want to apply it in this way. So is that something that is a significant social risk? Should we be thinking about prohibitions? Should we just let 100 flowers bloom? Anyone?
I think that one of the things that we tend to marginalize this topic by saying this is enhancement, and that's different from fixing choosing someone who will have Tay-Sachs disease or something like that. But when you talk about enhancement, if it actually adds quality years to their life, and as long as our society continues to have the behavior where we add quality years to people's life because they're wealthier, they marry better, whatever it is that causes your athletic, mental, and attractiveness to, then that, you can't say that it's completely free of health consequences. It's pretty close to not necessarily a good investment for society, but there are consequences.
In a certain way, I'm not so worried, because you didn't complete the whole thing. In order to do this in vitro fertilization, you pull off this cell and and then you reimplant the embryo in the mother. And it means not having babies the normal way. I actually think, for the most part, sex is likely to be the preferred way to have babies. And no, I think it is. I mean, because to have a serious social issue here, it would have to be that most people would choose to have their babies in test tubes rather than have them in other ways. So that's one. The question in my mind is, who's making the choice? Look, some of this will happen. We already have sex selection that goes on, aborting female fetuses, which I find abhorrent. But the question is, who makes these choices? Do we outlaw individual couples doing it? I don't know. I'm actually more concerned on the other side about the possibility of serious genetic diseases that are going to cost society a great deal. We might say, look, you're obviously a carrier of this serious genetic disease. It's going to cost a lot. You're free to choose to have a child that carries this genetic disease, but if you choose to make that choice, we're not going to pay for it. We will pay for in vitro fertilization. One may, if you put the choice in one set of hands, say the choice in the hands of the government, end up with something that could be sort of coercive. If I had to make a choice, I'd rather leave it in the hands of many, many parents, because I think on average parents will kind of do about the right things. They'll make some choices that I might not approve of. What I really would not like to do is centralize those choices and make them the subject of cost-benefit analyses, which is not entirely, a crazy outcome, that it could happen.
One interesting possibility, I haven't consulted with a lot of people on this, but a side consequence of the GINA passing could be that people start gaming the system and getting health insurance for their baby that they know is going to develop this disease because they didn't want to go through the in vitro fertilization. And then the response to the insurance company would have to be to put a lid on everybody because they can't discriminate anymore. And that means, I think that's as far as I can think through that particular issue.
So, here's a more social question. What effect will widespread genome sequencing have on personal identity, particularly in relation to ethnic or family identity? This is not a trivial issue. It is already possible to take someone's DNA and with actually a small number of the variants that Eric talked about, really quite accurately place them in terms of their geographic ancestry, what would popularly be called race. And this issue is not going to go away as we learn more about genetic variation.
I can say this is one of the most popular reasons why people do any kind of DNA testing right now as consumers. It's the basis for the Genographic Project, a lot of 23andMe, AfricanAncestry.com, and so forth. And then non-DNA based ancestry is a very big business. People are very excited about it, and they get interested when an African American, for example, has European ancestry and vice versa. So it will change people's opinion about these things that are mostly culturally based and based on what you've been told by your grandparents and so forth. So whether it will continue to do that for many, many generations after this sets in is another question.
But don't you think that people understanding that the complexity of your genetic inheritance versus, say, a black and white view of ethnicity is a positive thing?
I think it's very positive that they understand the nuances, but there's sometimes where it actually has economic impact. For example, if you can prove you're Native American, it could be 1/16th, 1/32nd, I think is where the cutoff is. It has impact.
I think the positive aspects you're pointing to are very important, and they really get to the heart of when a technology revolution happens, society digests it in some way, comes up with metaphors to understand it. You can take that genetic information about ethnic variation and draw maps that partition groups and say, I've seen genetic representations where you have trees, like evolutionary trees, and you have different groups coming off these trees that emphasizes the difference. You can also take people's genomes and lay them out and say that at this spot on chromosome 5, this guy from Japan is actually more closely related to this guy from Sweden than he is to this person from Japan. And that there's this tapestry of relatedness that varies at every spot in your genome. And the metaphors we choose and the representations we choose, also whether we refer to things as genetic variants or genetic defects, all these kind of words and images are up for grabs in this decade or so. And then, like our imagery of atomic bombs and other things, we settle in on some sets of social metaphors. And I think it's going to be very interesting which sets of images and words we choose for it, because it's not going to be without consequence.
People in the autistic community or in families affected with other diseases are scared that their genes will be selected against. What can you say to allay their fears? So I would just paraphrase that question. It's a broad concern in many communities that are affected by various types of disability, that as we strive to eliminate sort of defects, this is the quest for the perfect child, that we make it life more difficult in society for people who live with various types of disabilities.
Well, ironically, that's what we were just talking about during the break, was the tendency of society to try to make children conform to the same model for many decades after they're born. And so we're actually trying to change a healthy bell curve into something that's much narrower than is natural. And society may be losing some of its truly unusual and creative individuals by that, not just at the genetic level, but at every level, because of the messages we send out, that if you don't look like the models on the covers of the magazine with exactly their BMI and so forth, if you don't sit still in class and so forth. And so this will almost certainly ramify when genetics gets into the picture. And it's, you know.
But it's hard to say that variety in and of itself is the good. I mean, then you'd argue for more radiation, or?
Some have made the case.
Well, I think this question is from the autistic Asperger's community, and there are some arguments that mildly affected individuals do have something to offer society. We don't have to irradiate to find them, but we might want to encourage our educational systems to embrace them. But then you have the problem that there are some that are severely affected, which will be part of this diversity.
Take manic depression.
Yeah.
There is a long-standing notion of correlation between mild mania and creativity, of many creative people in the arts and the sciences being somewhat manic depressive. Certainly, experience in universities bears this out as well in a serious way. We could decide that genes, and manic depression can be an extremely serious disease, and it can be a disease that leads to suicide. What would you do if you had an option between implanting the kid who might have some predisposition toward that and could end up being a highly creative kid because of it? Do we call this a defect? Do we call this a potentially creative kid? We could do a study that says this genetic variant is correlated with manic depression, or we could possibly do a study that says it's correlated with creativity. And it might be the same variant.
But aren't we here fixed kind of with the thinking that there is nothing you can do with the bipolar disease and with your suicidal? Isn't the whole goal of our research that we would learn the pathways and cells of the brain causing this? The whole goal of this genetic research is not to deliver diagnostics for traits, which we barely understand what they are. The whole goal is that we would figure out what goes wrong in cells and tissues, and we could have, somehow interfere.
Now, and you bring us back to exactly the point I was trying to make at the beginning, that the goal is to understand the basis of phenotypes and the basis of disease, to have the power to intervene. The challenging thing may be the next several decades, when our ability to correlate exceeds our ability to intervene in any way, and we may go down certain paths, but I agree in the long run, we should be,
And we take these tests, or rather companies, we as a society take these tests too early out. And we really, without detailed studies, without understanding that the information of the genetic test is completely different if the test is carried out for somebody who is 5 years old or 75 years old. So without understanding the whole crosstalk between genes and environment, and we go out and launch these tests. So there are these moral or ethical issues are amplified by that.
If you went in, everybody's had their kids, but put yourself back to childbearing age. If you went in and you got the checklist now of 1,000 things you could ask for, which ones would you be asking for? What would you want to know about a baby? The amniocentesis will pick up more than Downs, let's say 5 years from now. What's on your checklist that which box would you check yes? I assume that you're going to somehow know enough about these 1,000 boxes to know which ones to check yes next to.
The key box to check is not to let the child be born in a poor country.
Yep.
Exactly.
Yep.
Exactly.
Exactly.
Any other comments?
No, that environment has a
That is
far
environment.
bigger effect on the child's outcome than anything that we can
Yeah,
possibly
certainly in
say.
terms of their health.
In terms of health,
Yeah.
in terms of all opportunity.
Right.
For everything we say about genes, of course, environment plays such a huge role in the fulfillment of human potential. People talk about genes for intelligence, genes for this. Just compare society 500 years ago, society 100 years ago, or societies around the world today. The gene pools aren't that all different.
Mm-hmm.
The outcomes are fabulously different. And that is the strongest argument against the naive genetic determinism
Right.
that you're going to have. Because we know what the influence of the social environment and economic environment which you grow up is.
So there have been several questions about education. And here's one from Max and Phyllis. Simply states, how do we best create a truly well-informed public about genetics? There's obviously concern, abstracting from various other questions, that we're pushing a lot of decision-making about relatively technical issues out into the public, and what are we going to do to try to make them better educated, especially sort of beyond the confines of Kane Hall, which is not a fair representative of the educational challenge we face.
I think one thing that you mentioned earlier is participation. There is, if you just lecture at people or just provide books, it's not the same as if they actually think that they can directly benefit and they're actually participating in the decision making. So I think they can either get involved in research or in community efforts. These various personal genomics companies that Eric showed may not be that effective at medicine. They may be fairly effective at ancestry, but they might be quite effective at education, where one person getting their $1,000 worth of DNA actually educates the whole family around them, in which case it's like buying a nice meal for everybody in your family, and it's not really that, or a nice education for everybody in your family. And I think we need to explore these different options, which are much less passive than say, an undergraduate education in genetics, which is also a good thing.
But don't you think a lot of this sequencing today gets people worrying about diseases that are so rare that just a moment's worrying about them is worse than the overall burden of disease?
Well, this is actually a question that often comes up, and there have been some social studies on this, and it turns out that people respond overall fairly well. I mean, they do some things that sound irrational, like they exercise more, even though it won't have any effect. But it's not bad, really.
No, I mean, just even looking at that catalog of those diseases depressed me. I put it down immediately.
I find it very anxiety-provoking. I don't want to know any of this. I like to study it, but I don't want to know any of this about me.
Yeah, I think it's going to be different. Some people like red hot convertibles and other people like Volvos. You know, it's just really it will vary.
Yeah,
And
but
that's
that-
mappable too, you know, right?
But that said, we shouldn't leave the education to these private companies educating about the diseases. What I think we need as a community is some kind of more efficient cataloging system for all the variants. In the sense Eric was mentioning, we have these Excel sheets for 1 million variants. But what we don't have is, we are very good in producing alignments of the sequence, but we are not very good in providing for public scientifically solid evidence for the functional variants, actually, so that we would have something like health code in the web where people actually could, which would be renewable, and it will be complemented monthly or weekly or whatever, but then people really could have a solid basis for their judgment that how useful is actually the 23andMe.
No, but what happens when we get everybody's genome sequenced? It gets cheap enough that people get their genome sequenced. The common variants, you'll build up a big statistical pile of evidence and you'll be able to interpret them. But there will be a million variants, there'll be several 100,000 variants that are rare genetic variants that there's no database on, never have been. And we'll be able to say, to Bill's point about anxiety provoking, this gene here has a non-synonymous change of an amino acid. It's never been seen before. But it is known that severe mutations in that gene have some very deleterious effect. We can't be sure that this means anything, but you know, you might want to think about it. You might want to worry about it. We're going to have like a long list of things that are sufficiently rare that we can't possibly interpret them, and they will basically multiply 10 to the 5th such things by the minimal anxiety you can feel about all of them. And the point about maybe it actually isn't worth knowing all that.
Yeah, this,
That I buy, but there should be a systematic cataloging effort to
Oh yeah, no, to
report
get
back.
the meaningful ones, yeah.
There's certainly a concern that we're going to add to what modern medicine is already very good at, and that is creating the so-called worried well. We have a society of the worried well, and now they'll have some new things to worry about, but we hope that they'll stay well while they're worrying. So here's a relatively complicated question from Lindsay about insurance. It's a sort of a different angle to look at it. Let me just paraphrase it by saying that, let's suppose, for example, that genome sequencing gets cheap enough that it is kind of standard of care for people to be offered the option to have their genome sequenced, and that it's paid for by insurance. Should the insurance company, under such circumstances, be able to look at the genome sequence even if they are legally prohibited from discriminating on the basis of it?
What are they going to do with it? Just curiosity?
Oh, it might greatly improve their ability to develop actuarial tables, for example, if they had all of these data. And then they could use that information for standard insurance purposes. I think the question is, it's an interesting one, is to how much are we worried about privacy and how much are we worried about discrimination? They're related issues, but the point here, I believe, is that they have a distinct way of playing out in society.
Well, the issue of privacy in your health record is already this huge one. So far, it's just generated more paperwork, but there's lots of companies, including Microsoft, working on making it so you get the efficiency of digital sharing without giving up the privacy. It is interesting that some people may want to see their genome if they could be assured that only they would get to see it and it doesn't become some broadly public thing. I mean, you'd wonder, later will they discover, does the genome predict intelligence or marriageability or something that if you reveal your genome and then they do this analysis, you say, well, I want it back. Well, that's pretty difficult to go and get it. I mean, the insurance company, if they could, there's certain preventative measures that you might want to know. And saying these things without any probabilities in mind, say, you should avoid certain foods. Boy, you'd want your insurance company to at least, even if they didn't charge you a different amount, because if you eat it, something terrible can happen. But you'd like them to tell you about that. So you can certainly create cases where the cause and effect relationship is so strong that people really should have the information. Right. Certain testing, we encourage people to get tests, and that's because we've done this trade-off that we think it's a valuable thing. If you go out 10 and 20 years, a lot of this data, I think, will become actionable. Don't you think?
Yeah.
Right.
Yeah.
So there is a question here, for example, about do we expect this to permeate the legal system as, for example, a defense? I got bad sequences, and
I'm willing to bet that somebody will try it. I don't think the courts will have a lot
What's
of
happened with
patience
sex
for it.
chromosome already? There's some
Yeah,
unusual
exactly.
sex chromosomes
That's
that have come
right.
up in
XYY
criminal
and
cases.
XYY.
Yeah.
That's right.
it's a close relative of a question I have about what are the prospects of genetics being used to predict personalities, antisocial behavior, so forth?
I think it will, actually. We already know that temperament is very genetically defined, and the first gene findings are coming out. So, there are certain features in personality which are definitely extremely heritable, and it doesn't prevent you from developing a character, but there are many, many heritable elements in those traits which we are kind of cautious that we don't come up and say that, yes, there are personality traits which are definitely can be predicted.
Do you think that personal genomics is finally going to answer some of those questions, or will the pendulum just keep swinging?
I think what it's going to allow us to do is to realize that it's a huge combination of those two, and we might feel that we can control both of them, rather than right now we're just controlling one of them. And whether we want to control both of them is another question. So for example, phenylketonuria is one of the things that every single baby that's born in a hospital, almost 4 million a year in the United States, is tested for PKU. You could call it an intelligence retardation gene. It's 100% heritable if you don't treat it. It's 100% environmental if you do. So it's already, I think we already have some idea about the nuance. We just need lots and lots more examples where we can exert the right environment to see if we can get closer to 100% environmental influence.
Yeah, this is where the diversity questions come in. So when you get into personalities and mental traits, I won't get this quite right, but imagine that there are genetic influences on your reaction time on tests. That wouldn't shock me, neurotransmitter differences, or
Age is the biggest.
age is a big one. That's right. That's right. But the relevant age that they're going to care about is in school age there, or attention spans or other things. How well a kid succeeds, given the particular dispositions you have toward what kinds of memory, what kinds of attention, what kind of reaction times, is going to depend a tremendous amount on the school system. In some school systems, where a kid who gets a chance to explore things they're interested in exploring, the kid who has a different type of attention span may succeed better, and in others they may not. And when we do in some kind of crude way an association between some variant associated with some core mental process, like a neurotransmitter, and we try to say, well, that's associated with good performance in school, what we may really be saying is good performance in a particular type of school system that channels you in a particular type of way. And so here, this isn't the namby-pamby kind of diversity. We need many kinds of diverse thinkers. And I don't think it's going to be the case that there is a best way to approach thinking problems. And yet, we still have a society that thinks about IQ as a one-dimensional linear scale. People are still pretty unsophisticated about different kinds of intelligences. And yet, I always like to think, if you got Americans to argue about not intelligence quotient, but athletic quotient, everyone would instantly realize that you can't compare different kinds of athletes because they're obviously on different scales. And yet for intelligence, we don't think that way. And we ought to.
So what does the public's fascination with genetics, what risk does it pose to distracting people from more important public health, and one might say
Enormous.
educational and other issues,
Look how many people we got out.
such as eating right,
Such
such as eating right. So neglect of these issues is already killing thousands. So a low estimate, I'm quite confident.
Not eating is killing more than not eating right.
Certainly our enterprise is frequently criticized in the political system, the NIH system, for just overinvesting in genetics and genomics as opposed to other more important public health issues.
Well, I certainly think genomics in general is the key to solving virtually all health problems, whether it's infectious disease or cancer or anything. So this argument about what you do with the individual information is important, but no way, no matter what your position is on that, the idea that we're gaining a broad understanding of how the human body works, that's going to allow us to make huge advances in medicine. It is ironic, the period since 2001 has been a particularly bad period in terms of new drug discovery, which I think most people, if you'd asked them in 2001, they would have said, okay, here we go. But I think that's just other factors that create that. We will reap the benefit of these things in the years ahead.
Implicit in much of this discussion is all new technologies that have broad implications raise new fears and real risks in society, and are we sort of overreacting to the risk-fear side of genetics and underappreciating its potential to lead toward the world that Bill articulated?
Well, almost every technology we've been afraid of, and so far it's all worked out pretty well. I mean, cars kill 50,000 people a year, but still people like them. Computers give you a lot of spam, but still people like them.
It doesn't kill you.
So many, fortunately, we haven't run into one yet where we really caused a problem. It doesn't mean it's not a good thing to always think, oh, the next one is the one that we really got to think through carefully, talk to people broadly. It's a little too easy to say, hey, we haven't come up with a bad technology yet. Putting nuclear weaponry is one that we haven't used badly yet. But so I think you can be somewhat optimistic about that.
But particularly in this case, as you said, it's about understanding fundamental mechanisms. As much as we're here talking about the personal genome and your own information, that isn't why any of us doing genomics are doing this. It isn't about assigning individual risk. What excites everybody, what we all come to work for in the morning, is the chance to finally find out what is the mechanism underlying diabetes or schizophrenia. It's to open those black boxes and figure it's going to lead to new medications, new diagnostics that can be used in support of those medications. And I don't think it's about personal assignment of risk for most of us. So as a technology, when I weigh whatever risks there might be, if I look at it and I say, how could we tell our kids we didn't go and find the actual basis of disease because we were afraid that you might misuse the information? I don't think it's even a close call.
Here's a pointed one. Have any of the panelists had their genomes sequenced? Will you? If so, what did you learn, or do you hope to learn?
Start at that end.
Maybe we should go the other way, because I think George is the most exposed on this question. Eric.
I have not had my genome sequenced. We actually discussed this a little bit before. Not had my genome sequenced. I have no burning desire to get my genome sequenced because there's no particular thing that I want to know for which it is the answer. But I did note when we talked about it over dinner beforehand that if, God forbid, I had a cancer, I'd get my cancer sequenced in a heartbeat and compare it to my normal genome to look at whatever I could learn, because I think there would be something I could imagine doing with it. So for me, it's very much tied to it being the answer to a relevant question.
There is no female sequenced so far, so no. That said, I wouldn't like to have it sequenced. Well, I have no strong opinions. I don't mind if it's sequenced, but let's put it that way. I have been scanned for, I come from Finland, and there is a cluster of some 40 diseases which are more prevalent. Monogenic, severe diseases, more prevalent in Finland than anywhere else in the world. So I have been scanned for carriership for all those mutations.
I did Tay-Sachs.
And
Yeah.
I'm an Ashkenazi Jew,
Right.
by the answer.
Exactly.
I did Tay-Sachs. That's relevant.
Okay. So there is some value for those diseases.
Oh, definitely.
Tay-Sachs and Finnish
Oh, yeah,
specific
carrier
disease, yeah.
screening, definitely.
And I said at dinner, after we cure the top 20 infectious diseases, then I'll get my gene sequenced.
Bill gave us this challenge at dinner that if we, he didn't tell us whether he was going to reveal his sequence, but how about that? Is that a deal if we develop cures for the top 20 infectious diseases in the world today, that you'll have your genome sequenced and reveal it to us?
Absolutely.
Great.
Right.
Now we come to George.
So I'm part of, I'm a subject as well as a director of the Personal Genome Project, and my IRB, Institutional Review Board, actually requested that I be in the initial cohort so that I would experience all the risks and benefits firsthand and understand the issues. And it has been a learning experience. We are not yet announcing that we have a little bit of genetic data about everybody. We're going to try to get everybody in one group rather than making it about one person. And that should be very soon. And it will have some female and some African American components in it. And we hope to expand that quite a bit in the coming years. So I haven't learned anything worthy of noting, and don't expect to learn anything. I think it'll be like the Finnish diseases and the Ashkenazi diseases. I hope to get no information. No news would be good news as far as I'm concerned. I know that I will have narcolepsy in there for sure.
And what about the issue of privacy of the information?
Again, I think there's a broad bell curve of importance of privacy to people, and the people that are signing up for our study are acknowledging that we may not be able to keep a secret even if we wanted to. For example, 26 million veterans' records were revealed somewhat innocently. 15-year-old kid can identify his father from just a cheek swab and a database of surnames. So that's part of the privacy issue as far as we're concerned. But the other aspect is we want to use as a vehicle for getting people up to speed enough so they can really make an intelligent decision as to whether they want to participate in any of these sort of studies, knowing what the consequences might be way off in the future where we don't know where things are going.
Sort of representing the questioner's intent, I just wanted you to comment on your own personal view about this privacy, the importance to you of maintaining the privacy of the data.
Okay. I don't think I'm on the high extreme. I'm sort of in Bill's category. I'd be happy to be part of a movement, if there is one, to revealing. We're writing the book as we go along. So, for example, right now, people in general reveal their face. In a way, right now, a face is more revealing than your genome. A small minority of the population doesn't go outside or wears clothing that covers their face, but basically it reveals whether you're sick enough to show it on your face, whether you're angry or happy, and various other things that are really quite revealing. And it's just a social mores that's to set that standard. And the genomics, we don't know what the standard is going to be. It could be mandatory, as Eric set up for presidents, the way, you know, this SEC rules for revealing salaries are different from what it used to be, and it may vary from culture to culture. So I don't know where it's going, but I think I fall slightly on the end of I'm willing to reveal that information if it's value to science or some other value.
All good things have to come to an end. I'll turn this back over to Bob. I think this has been really fascinating and illuminating, and seeing some of the questions I couldn't get here today. I think we should start a collective blog on this subject, and I suspect that the conversation could go on and on and on. So thank you.